thank you very much to the organizers and the people who've put a lot of work into this. And I've seen Amy Kyle working day and night, and I'm sure a lot of other people behind the scenes. So I think it's a terrific and very stimulating morning that we've had, and afternoon as well. Okay, so what is autism? I'm assuming that at least some people in the room maybe aren't as familiar with autism. So it was first identified in 1943 by a psychiatrist named Leo Kanner, who had seen a series of children come through his practice, 11 of them. He wrote this up in his seminal paper, and he described and he coined the expression autism for this condition that involved 3 domains of deficit, the first being social reciprocity, the second being this lack of communicative skills, and then a pattern, a sort of ritualistic type of behavior, repetitive behaviors, or restricted interests. And the definitions that we use today are that these symptoms are present by 36 months of age, and it's considered to be lifelong, but this is actually a bit controversial, and there's certainly anecdotally, there do appear to be people who outgrow it, quote unquote, and maybe later in life really don't appear particularly odd or different at all. So autism falls under this umbrella called pervasive developmental disorders, which includes all 5 of the conditions shown here, and 3 of them are considered the smaller umbrella of autism spectrum disorders. Also, you should know that the new definition based on what psychiatrists use, which is the Diagnostic and Statistical Manual for Mental Health Conditions, in the new edition, which just went into effect, I think, in the last week or so, there is actually no distinction among those 3 that are in the autism spectrum. So autistic disorder, Asperger's, and the PDD-NOS will all be one. There will not be a distinction anymore. The diagnosis, these are the 2 gold standard instruments used for diagnosing autism. The first is usually referred to as ADOS, for Autism Diagnostic Observation Scale. And this is a standardized but semi-structured play interaction with a child. There are presses that are designed to evoke certain kinds of responses, social and that sort of thing. And I'll play a short clip of one of these. It's a short 20 or 30 minute kind of exam. And then the other is the Autism Diagnostic Inventory Revised, or ADIR. And this is a fairly lengthy interview with the primary caregiver. So this is what gives you kind of the history of the development, what's been happening with the child from birth to the time when the interview takes place. The other one is more of that one-time snapshot. And then both of these provide scores in each of the domains, those 3 domains. So I'm going to play a clip here, and this is an examiner. Leslie has just told the baby, oh, let's have a birthday party. It's baby's birthday. This is the baby here, the little doll.
Now we have to light the candles. Ready? Let's light them. Oh, it's hot. Oh, hot. Ready? Let's see.
And you notice he's ignoring her totally. He's in his own world.
Happy birthday to you, baby. Happy birthday to you.
Trying to get him to sing. She's going to try and sing this thing. Blow out the candles.
Okay, time for cake.
He didn't
Baby's
participate
so
in blowing
hungry.
candles. A child at 3 would be doing that.
Baby's so hungry.
So now she's going to try to engage him in
Time for
actually
cake.
taking a piece of the cake to feed the baby, which is what a child at this age would do in
Can you
a
give
play
baby
situation.
some cake? She's hungry.
So he's digging in with his little thing to get a piece.
Here you go, baby.
She demonstrates.
Here you go.
Can feed the baby. He's picking up the cake, the Play-Doh there. Oops, he dropped a piece. Bends down to pick it up. When he gets up, and he doesn't actually feed it to the baby. Actually, you'll see, he tries to put it in his own mouth.
Oh,
so
no, not for you,
he hasn't
honey.
gotten the idea here. This is not a real cake.
Not for you. Oh.
Okay, so this child actually did have a diagnosis of autism, and you also might have noticed he didn't make eye contact. He never really looked up at the examiner. There was really no social interaction going on at all. Some facts about autism: it has a sex ratio of 4 to 1. The prevalence in the United States is about 1 in 88 by the latest CDC surveillance figure, which comes to 1 in 58 for boys. Historically, autism for several decades was attributed to a cold parenting style, and this really arose from work by Bruno Bettelheim, who published in the 1960s, saying that essentially this was really parents who were responsible for this kind of turning inward of children. We know that that is actually truly not the case, that there is a neurobiologic basis, there's a pathophysiologic phenomenon going on, and you can see it with postmortem autopsy studies that show a lot of lesions in the brain, everything from a fuzzy separation between gray and white matter to a loss of Purkinje cells, and then there's electrophysiologic evidence. MRI and fMRI have given us even more clues about aberrant brain functioning. And that is clearly the case. And I think it is also important to know, and this sort of has to do with how scientists relate to communities, which is that there is this sense in the autism community that the scientific community at heart still blames them. And I would say that, it still colors what goes on. And there are many parents who have certain ideas about what is causing autism in their child. And I think it's really important to engage those people, regardless of what we might think of their specific hypotheses. And I think there's too much dismissing of parents who happen to have one set of beliefs. What causes autism? Well, definitely genes play a role, and there have been many twin studies going back for several decades, and also environmental factors do appear to play a role. The largest, there are actually 2 really large studies of twins recently that have shown heritability estimates going from around 38% to maybe a little bit higher than that. Previously, and you'll still hear a lot of experts quoting the older studies, usually they were small studies, 10, 15, 25 people, or pairs of twins, that came up with estimates of 70 to 90%. So I think the larger studies are really showing it's not as heavily genetic, even though clearly there is a genetic component, and probably there's a lot of genetic environment interactions, and I'll talk a little bit about some examples of that. But this is important that it is environmental, and this, of course, I was preaching totally to the choir, but this is important because, for public health reasons, the environment we can intervene on, the genetics, not so easily. So, this is kind of an assessment I did 2 years ago. I think it's still, so these are many of the candidate suspects that are out there, genes for sure, and that world is continuing to look further, and they're finding that it's a very complicated genetic picture, not as simple as, in 2003 I was in meetings where these people were saying to the NIH roadmap people, we're going to have all the genetics figured out in 5 to 10 years. We'll have it all figured out. We've got GWAS now, it'll solve the whole thing. Environmental chemicals, I'll talk about several chemicals that we've looked at. There have been studies, for example, on pesticides, which I won't be touching on later, but I just want to say that the Chamaco study actually had one of the very first papers suggesting that pervasive developmental delay symptoms are correlated with the organophosphate pesticides. Another study was done in the Central Valley by Eric Roberts using the pesticide use report system, the agricultural applications. Chamaco was measuring urinary metabolites in the mothers. So the pesticide applications that Roberts looked at, it was the organochlorines that seemed to rise to having a strong association. There are also issues related to vitamins and nutrients. I'll give some report to you a little bit on that. Microbiological agents have been of interest, and in fact, some of probably the very first, at least that I know of, environmental factor that was published came in the 1970s when there was an epidemic of the rubella virus in several parts of the United States, and there were 2 papers, 2 different case series suggesting pretty high rate of autistic symptoms in those children who were congenitally exposed to rubella. And there are other hypotheses around influenza. And it may actually be not so much the infection, but perhaps the response to the infection. And so we actually published some data showing that fever seemed to be, a fever associated with influenza or other causes seems to have some association. Then there are the gestational and perinatal sort of conditions, and some data beginning to emerge around that. Fertility treatments have been hypothesized because of perhaps the trend, paralleling trends in autism incidence. Other medical interventions, I think, are of interest, such as ultrasound, medications, certainly, and then maybe maternal reproductive characteristics as well. And then, modern technology. People have been hypothesizing cell phones do various things, and these are questions yet to be determined for many of these factors. So just a word about time trends, because I know this is there's a lot of controversy about the trends. And we looked at the California Department of Developmental Services data, which covers the whole state. We have a system of regional centers that provide services. And we found that the autism incidence up to 5 years of age, from the 1990 births to the 2001 births, actually rose sevenfold, which is a 600% increase. And when we tried to attribute what that might have been due to from the point of view of just better awareness and changes in diagnostic criteria and so forth, we came up with about 200 of that 600% increase that we could explain through these artifacts, let's say, of diagnosis and reporting, and also some changes like younger age at diagnosis or older ages of mothers at childbearing. Given that it really looks like at least part of this increase doesn't get explained so easily, there is a possibility of a true increase. And I just think it's a good thought experiment. What are the other things that are rising in this same period of the last few decades? And is there some sort of a common unifying explanation that might link these in some way, that these may have some common causes or common kinds of mechanisms that are operating? So now, in the remainder, I want to talk to you about some of the results from the study that we launched just about 10 years ago, called Childhood Autism Risks from Genetics and the Environment, the CHARGE study, which had as its goals to determine what are the environmental and genetic causes and contributing factors, and what about the interactions of those two. And then a second goal was to see if there might be subsets of cases of autism that were characterized either through behavioral differences or immunologic differences or genomic or metabolomic ones that actually etiologically would be linked, that certain kinds of causal pathways or causal exposures might particularly produce certain types of autism. And in planning the study, we looked at the literature, and there was almost nothing published. The rubella paper, and really, environmentally speaking, not a lot out there. So we say, well, what are the neurodevelopmental toxins that we know about? Well, pesticides, metals, some of those persistent organics, the viruses and infections we wanted to also take on, and then medical procedures and nutritional factors. And then, these are ways that we can collect either direct exposure data or information about potential exposures using biospecimens of various types, conducting interviews, linking to exposure databases, and that comes from having the residential information. And of course, we heard about the pesticide use report, but there's many interesting exposure databases in California. And then, of course, medical records is another way to get at past exposures. And so just there, for any one thing we're interested in, there may be multiple ways to get at that exposure. So the basic design was a case control with population-based recruitment, which by and large, had not been done when we launched this study, and including standardized clinical confirmation of the diagnoses. And then in addition, this was part of our children's center, so we were linked to these laboratories that had, state-of-the-art equipment and methodologies worked out to look at biomarkers and exposures. So we have 3 groups. Usually case control, it's 2, but in this case we have children with autism and children with developmental delay, because we were interested to know whether the associations we might find would be specific to autism, or might they actually be really impacting neurodevelopment, and for reasons we don't at this point understand, some children might go in the direction of developmental delay, some might go in the direction of the social issues associated with autism, and some might actually have both of those problems. And then the controls come from the birth files, and these are frequency matched to our projected distributions in the cases of age, gender, and geography. And we've had a great collaboration with the DDS on this. So these are the eligibility criteria for all of the 3 groups. They do have to be born in California, that's so that we can get their newborn blood spots, and live with a biologic parent and reside in the catchment area, and the parent speaks either English or Spanish. The child assessment, we do that ADOS, we do the ADIR, we do a medical exam, we get history in the child and the family. And also a broad assessment, we do cognitive and adaptive behavior scales as well. Again, they come in from those 3 groups, but then we do this full diagnostic assessment so that we place them in the right category. We have kids who come in from the general population, but they actually meet criteria for developmental delay. We have some that come in as developmentally delayed, but we actually find that they meet criteria for autism. So we do this complete assessment and then assign a confirmed diagnosis. And then, of course, we collect a lot of exposure data through interviews, and we collect specimens, and we get permission to obtain these different kinds of medical charts here. And so our slogan and what we recommend to the families we meet is it's time to be in charge. And moving to the results, we start with metals. The goal, this is the first project looking at metals, was to determine whether the current blood mercury levels differed in children with autism or ASD compared with the typically developing controls. Now, I just want to explain what we were doing here. So this was in part because there were many families who felt that their children were overloaded with toxins, that the children were not metabolizing very well. And there had been some anecdotal sorts of reports about how high the levels were. A lot of parents were coming to us, including those who were on our community advisory board, and saying that you should be recommending every child be chelated, because I had my child chelated and tons of metals came out. So we really wanted to see in an unbiased way, do these children seem to have really high levels? So this is just the crude histogram, and if you look at it closely, you'll see that I've got the blue line, which is, I think, the overall mean, and this is on a log scale because, of course, it's a little bit skewed out to the high end, towards the low end, with some tail on the high end, but the peak of the typicals, you can see, is a bit higher than the peak of the autism and the developmental delay group. So in fact, at first blush, it looks like actually those children have lower levels of mercury circulating than the others. But then we went a little further and we said, well, why might that be the case? And in fact, I think it was even significant, this difference, that they were lower. Turns out that they didn't eat nearly as much fish, and children with autism are known to be picky eaters. Turned out they ate less fish, they ate fewer fish meals per week if they did eat fish. And when we adjusted for their fish consumption in a model looking at their diagnosis as a predictor of their blood mercury level, we saw that there's really no difference between the typicals and the autism group, with no significance. The difference with 4% difference. There still was a lower level in the developmental delay group, which probably deserves some thought as to what might be going on there. Are there behaviors besides the eating issue leading to that? Very strong contribution, as you can see, from all three types of fish, which we got detailed information on. Also, children who had mercury amalgams, and these were young kids, 2 to 5 years of age. A lot of them haven't had that much dental problems yet, but those who had the amalgams and also either chewed gum or had a habit of grinding their teeth actually had higher levels of mercury. If they had either one of those alone, it didn't make a difference. But if they had both of those characteristics. So bottom line, children with ASD do not have higher blood mercury. I think that's an important message, that after we adjust for the fish and other mercury sources, there really was no difference. Now this, I have to give you the caveat, of course, this does not address the question mercury is a causal factor, because circulating mercury levels, the methylmercury has about a 70 to 90 day half-life, and these children, probably took them a couple months after they got the diagnosis till we got their names, and by the time we called them, got them into the clinic, the mercury we're looking at is definitely post-diagnosis mercury. And so we are undertaking a study using baby hair that mothers bring in if they've saved the first haircut, and newborn blood spots, and I don't have those results. Air pollution was a second factor. We were interested in finding out whether mothers of cases were more likely to live near freeways or have higher exposures to ambient air pollutants as compared to the mothers of controls. And the way we did this was we characterized where their household was using ArcGIS to map the homes. And then using the Caltrans database on freeways, determine how close they were to their closest freeway, with the referent group being those who were 9/10 of a mile or further, basically 50% of the furthest 50% of the population. And those in the closest 10% had this higher odds ratio of about almost 1.9. You'll notice that there's not a dose response here. But in other words, all the other groups are closer to the referent group, which turns out to be consistent with exposure data that shows that particle concentrations around roadways is particularly high. Nearby, it drops by about 300 meters, which is actually about the cut point of that top 10%, back to about background. So maybe we wouldn't have expected to see a dose response given the distribution of our distances that we had. It's an exposure measure that is objective. It was not based on self-report, except report of where they lived, which we think people probably report pretty well where they lived. Proximity traffic has been associated with asthma. We know that many of the compounds that are in air and traffic-related air pollution have been shown to induce some sort of neurodevelopmental delays or deficits in rodent studies, and other kinds of mechanisms like oxidative stress, inflammation, endocrine disruption, and so forth could be going on. So the third factor I want to talk about is in relation to nutrition. And this is work done by Dr. Rebecca Schmidt, who came originally as a postdoc, and she's now on our faculty. And she looked at the intake of prenatal vitamin supplements. One of the components of those, one of the main components, not the only one, is folic acid, which is required for DNA synthesis or repair and methylation. Supplementation with folic acid prevents a high proportion of neural tube defects, and women are recommended to take these before and in the early period of pregnancy. And we have fortification of cereals and grains since 1998, and since then NTDs have actually declined in the U.S. And in her analysis, you can see that the mothers who took these prenatal vitamins in those early months, this is 3 months before pregnancy, -3, -2, -1, and the first month of pregnancy, you'll see a significant difference there for the case mothers and the control mothers. Control mothers were more likely to have taken them. So the odds ratio here was 0.6. In other words, this is a protective effect, approximately 40% lower risk in the people who took them. Rebecca went on to then analyze the full diet, the cereals and the grains and other sources, not just the prenatal supplements, and found that in overall higher intake was associated with lower risk for autism. Also in this, she looked at some gene-environment interactions, focusing on some of the genes that are responsible for this, conversion of folic acid to folate, and then the folate being a methyl donor. So I'm going to show you the results for the COMT gene from the child, in which those who, what you see here is really interesting, because those who had the low-risk genotype and who did supplement, that's the referent group. If you have the inefficient transmethylation gene here in the child, then there is a slight increase, 1.3, and similarly, if you did not take your prenatal supplements, but you had the efficient transmethylation, you're at 1.8. But if you happen to be in the disadvantaged group on both the genetics and the environment, we see a 7-fold increased risk for autism. So really a synergistic effect here. And I think this is probably the first that we know of an example of a gene-environment interaction in relation to autism. So I think it's interesting, even though I think most of the things OEHA is interested in, it's not so much the nutrition, but we have been talking about these other factors. And I think this is a really nice example that can be carried to look at the other types of environmental factors that we look at. And also she showed that maternal and child genes actually add additional risk. And then very quickly, I'll just talk about maternal metabolic conditions. The mother provides this intrauterine environment, and not only what she takes in from the outside, but also what's going on inside her body matters. And this is a schematic for 2 possible pathways by which insulin resistance could result in harm to the developing brain. And on the left side, it's more of an inflammatory pathway. On the right side, it's more of the direct hyperinsulinemia, leading to potentially fetal hypoxia. And these are conditions that could, in fact, alter that gestational brain development. And so this is a slide really just showing some of the results looking at diabetes, hypertension, and obesity, 3 conditions that are considered part of the metabolic syndrome. And you can see that the odds ratios are elevated. We have, the bottom line is having any one of these increased the risk by about 60%, but it also increased the risk for developmental delay. And so you can see that odds ratio is 2.5 for having any one of these conditions for developmental delay. So it's not specific to autism what we're seeing. And in fact, there have been prior studies suggesting that children of for example, diabetic mothers often have some cognitive deficits. Now, I think this goes back to this little thought experiment I brought up in the beginning, which is what are the causes for the maternal metabolic conditions? We're seeing, the increase in obesity. In fact, there are a number of studies out now, cross-sectional ones at this point, that do show associations between body burdens of a number of endocrine disruptors with diabetes and obesity, and including, for example, bisphenol A. Which has gotten a little bit of publicity. And so maybe this is part of the pathway by which some of our environmental factors are working through disruption in maternal metabolic regulation that is, in fact, then influencing the child through a pathway that maybe we hadn't considered before. And then the last point from our results I just want to raise, again, sort of broadening out our concept of causality here, that it may be direct effects on neurons is not necessarily what's going on. We have shown a lot of differences in terms of the immune markers in children with autism compared to their counterparts who are developing typically. These are some of the studies that have been published looking at things like the overall IgG, reduced levels of one of the cytokines, TGF-beta, that is regulatory in immune responses, abnormal natural killer cell functioning, elevated cytokines that actually correlate directly with not just the diagnosis but individual behaviors that we've looked at, and then leptin levels, which have been elevated in these children. And leptin has a number of different kinds of functions. And then this is a Western blot looking at maternal IgG antibodies to fetal brain tissue. And what you see is on the left side are the three autism moms, and you can see these arrows showing a couple of proteins that are, this antibody response is seen in the autism group, and this particular pattern is not seen in any of our typically developing moms. And the first paper we published, we had about 60 in each group. We've now confirmed this in hundreds of cases and hundreds of typically developing mothers of typically developing children who do not show this particular pattern. And of course, we don't know whether this is upstream of the child's aberrant brain development, or is it the other way around? Is the child brain development somehow influencing the maternal response, and the tolerance somehow has been broken here, it appears. But we do know that there's a lot of communication between immune cells and various cells in the CNS through neurotransmitters, cytokines, and chemokines, where the receptors of each of these are on the other system's cells. So the possibility is out there that environmental chemicals might influence neurodevelopment generally, autism specifically, through this immune dysregulation, a kind of indirect effect. And it really changed our thinking about autism as a brain disease as opposed to a whole body system, and that maybe factors that may not be thought of as neurotoxins may actually be able to influence these kinds of behavioral and cognitive development. So anyway, this, I think the significance of the CHARGE study is really, it is the first comprehensive study of environmental factors. Early studies had ecologic designs, were usually clinic-based if they didn't use an ecologic design, and sometimes don't have confirmation of the diagnosis. So those are some of the features of it that make it pretty unique, even 10 years later. And we are focused on what is modifiable because we really want to be able to intervene. And the rates of autism are going up. I'd like to see them plateau. I'd like to see them really start to come down. And I think that that's achievable. The CHARGE study, as sort of the final sort of caveat here, is the limitation is that it is retrospective. And although we have ways to try to get around that, that is a problem. And having prospective studies is truly important. That was highlighted by Asa and others today, Rosalind. And we have the MARBLE study, which is complementary to the CHARGE study, and it's pregnant moms who already have a child with autism whom we then follow forward in time, their pregnancies with lots of specimen collections, and then the child's development. And these eliminate the problem of differential reporting bias, and they permit the examination of, etiologically relevant, measurements that can be made in real time. These are some of the future studies that we have planned. I just want to mention a couple of these interaction issues that we've been looking at. Air pollution and the nutritional status of the mom as an interaction, going to the cumulative impact kind of place. I think we were starting at such a primitive place in our understanding when that the idea of cumulative impact was not on our minds 10 years ago, but now we can start thinking about that. And then just thanking all the collaborators and investigators from many places, some people in this room, our staff, and certainly the families who have been very, very generous with their time, and our funding agencies. So thank you very much for your time.